Welcome to the Advanced Fluorescence Microscopy and Image Processing course. So today is the fifth lecture. Today we will learn about 6-dimensional imaging, live cell imaging and time-lapse imaging. So let me start with 3-dimensional imaging. What is a 3-dimensional imaging and why we need to image a cell in 3 dimensions? So here you can see this is a cell, this is a glass slide and this is the objective. Consider that this is an inverted microscope, so objective is on the bottom and you are seeing the cell from the bottom. Now under the wide field illumination, what happens when you illuminate the sample, the illumination light or the excitation light illuminate the entire cell like this. And because of this, what will happen? Let's say there are 10 fluorescence molecule inside the cell. So all 10 molecules will give the fluorescence. But what will happen? The focal plane of this objective lens is only around 200 to 1000 nanometer in depth. Okay, so the objective can see only those molecules which are in this focal plane, and those are only 3 molecules out of 10. So what will happen? Even if the objective illuminates all the molecules, all the fluorescent molecules inside the cell, but it can capture the fluorescence coming from only these 3 molecules which are in the focal plane. And because of that, what will happen? Ideally, you are expecting an image like this in which you can see all 10 fluorescent molecules, but eventually you will get the image like this in which the 3 molecules which are in the focal plane, they are brighter, whereas the other molecules are out of focus. So how to collect this entire information? Let's say for your biological question, you want to count the number of spots inside the cell. And if you take this kind of image, you will be missing the spots which are above and below the focal plane. Okay, so how to collect the entire 3D volume of the cell? So to collect the entire 3D volume of the cell, what you have to do is you take one image at this focal plane, then second image at this focal plane, third image at this focal plane, fourth image at this focal plane, and fifth image at this focal plane. So you just move your slide little bit up and down so that the entire 3D volume of the cell is covered in 5 images. So this is known as Z-stacking, or the number of Z steps required to cover the entire cell volume is 5. So in 5 images, you can cover the entire Z volume of the cell. So this is known as 3D imaging. And after taking these 5 images, if you merge all of them, then you can see the image like this, which is known as the maximum intensity projection image. So you are projecting all the 5 stacks into one image. So this is a 2D representation of the 3D volume of the cell, and this image looks exactly like the ideal image that you were expecting. So when this Z stacking is required, so let's say if you are working with a virus or the bacterial cells and you are imaging the virus or the bacterial cells, at that time you do not need Z stacking, because you see the size of the virus is 200 nanometer, the size of the bacteria is 1 to 2 micron, and the depth of focus is 250 to 1000 nanometer. So virus and bacteria can be captured just in one focal plane. But to cover the to image the eukaryotic cell nucleus, which is 5 to 10 micrometer thick, okay, for imaging such kind of a thick sample, you need Z stackings to cover the entire 3D volume of the cell. But while imaging virus or bacteria, you do not need Z stacking. Okay. Now, how to avoid oversampling or undersampling while doing Z stacking? So let's say, suppose this is the cell and you want to get the entire 3D information from the cell. So you can image this cell by taking 5 images. But you can also image the cell by taking 8 images which are overlapping. Or also sometimes you may image the cell like this, in which some information is lost. So basically you have to define these steps to the microscope. You have to tell your microscope software that you have to take, let's say, 5 Z steps at every this distance, let's say 200 nanometer, 250 nanometer distance, to cover the entire Z volume of the cell. So how you decide, how you should decide this number of Z steps and the distance between the Z steps. Okay, at what distance the microscope should take this kind of multiple stacks, that you should you have to define. So let us see how you can decide that thing. So this is known as the desired sampling, because see, you can cover the entire cell volume in just 5 Z steps. So this is the desired sampling rate. But let's say if you take 8 images which are actually with overlapping focal planes, then it is known as oversampling. Now, what are the disadvantages of doing this? Because whatever the information that you can capture in just 5 steps, why you will go for taking 8 steps? Okay, that will lead to excessive photo bleaching and photo damage, because instead of exposing the cell for 5 times, you are exposing the cell to the light for 8 times. So it will create excessive photo bleaching and phototoxicity or the photo damage to the cell. Also, your file size will be larger, because earlier your file contains only 5 images, but now your file will contain 8 images. So your file size will be larger, and by making the larger file size, there is no gain of the information. It's not that here you could see 10 molecules and here you will be able to see 12 molecules. Okay, if there are only 10 molecules in the cell, here also you will see only 10 molecules in the maximum intensity projection image. Okay, but you will not get any additional information by oversampling. And obviously, taking more Z steps requires more time to cover the entire 3D volume of the cell. So these 5 images can be taken, let's say, in 1 minute, but microscope may take 1.5 minutes to take 8 Z sections. Okay. So these are the disadvantages of doing oversampling. Now, it is possible that if you do not know how to decide this sampling interval, then you may end up doing this kind of imaging, which is known as undersampling. So what are the disadvantages of undersampling? Loss of essential information. So here you can see the only 3 Z steps were taken, and in which some of the molecules are not captured. So you will end up with if your aim is to count this number of molecules, then you will end up with lower numbers than 10. Okay, and images cannot be used for quantification. So if you have captured your microscopic images by undersampling, those images you should not use for quantifying some biological information, for intensity measurement or for counting the number of spots per cell. For those kind of counting or quantification, you should not use this kind of imaging, because they are undersampled and the information is lost. So it is very essential to precisely sample your cell while doing the Z stacking. So it is recommended that image acquisition is done according to the Nyquist rate as much as possible. So Nyquist-Shannon sampling theorem. Okay, so this theorem states that when sampling a signal, the sampling frequency must be greater than twice the bandwidth of the input signal in order to be able to reconstruct the original signal from the sampled version. So let me explain you this sentence using this schematics. Okay, so this is a wave. Okay, and you want to capture this wave. So if you sample with this frequency, so this Fs stands for the sampling frequency, and this 2f stands for the frequency of the wave. Okay, so if the sampling frequency is less than 2 times of the wave frequency, then it will represent a faulty wave, because with this sampling frequency, what you will see that this wave is like this. Okay, you cannot see the real pattern of the wave. But here you can see the sampling frequency is equals to the wave frequency. So if you sample this wave at these positions, or maybe at these positions, then you can see the actual wave pattern of this signal. Okay, and this is an example of the oversampling. So the same wave, you can also get the information about this wave by oversampling it. If your sampling frequency is more than 2 times of the wave frequency, then it is considered as the oversampling. So these two conditions, where the sampling frequency is equivalent to the wave 2 times of the wave frequency, this is the perfect sampling. Okay, desired sampling. Here also in this diagram, you can see this is the adequately sampled signal, whereas this is aliased signal due to undersampling. So if you sample at only this point, this point, this point, this point, this point, then you will see this wave like this. you cannot see the real pattern of the wave. So your sampling frequency should be adequate to see this real wave pattern. So now I will show you how you can determine the z-step size for your imaging experiment. Okay, so for that you should use the Nyquist calculator. So click on this link and click on this Nyquist calculator. So you will see this kind of a Nyquist rate and PSF calculator. Okay. So here, to determine the z-step size, what you should do, you select the microscope type, whether you are using confocal microscope or the wide field microscope, spinning disk, or you are using STED or light sheet microscope, what kind of microscopy you are using. So let's say suppose you are using wide field microscopy. Then the numerical aperture of your system. So let's say the numerical aperture of your system is 1.4 and you are imaging a GFP protein. So our excitation wavelength is 488 nanometer and the emission wavelength is, let's say, 510 nanometer. Okay, so you have to input all these parameters and the number of excitation photons. So until unless you are using the 2-photon or multi-photon microscopy, you should keep this 1. Okay, for any kind of confocal or regular wide field microscopy, it will remain 1. But let's say if you are using multi-photon or a 2-photon microscopy, this number of excitation photon will be 2 or 3 depending on your application. But generally for confocal wide field imaging, this number will remain 1. And also you have to define your immersion medium. So let's say you are using an oil immersion objective lens. So for oil, the refractive index is 1.5. Suppose it's a glycerin immersion objective lens, then you have to select glycerin. So appropriate refractive index will come. So here I'll use, let's say, oil. Okay, and then I just have to press this button, calculate. So it will calculate and it will tell me X, Y, and Z sampling distance. Okay. Now today we will not discuss about this X and Y sampling distance, but it is very important. Okay, but that we will discuss in the class of camera technology. Okay, but today for our today's interest, this distance matters. The Z distance should be 272 nanometer. So you will tell your microscope software that it has to capture the images at every 272 nanometer to capture the entire Z volume of the cell. And number of Z sections depends on the size of your sample. So suppose you are imaging a 5 micrometer large cell, then the microscope will automatically determine that distance with this Z step size. So this is how you should check the Z step size using this Nyquist calculator before putting that value into the microscope. In these days, many modern microscopes automatically tells you these numbers based on the optics selected. So you may not need to use this calculator as such. For if you are working with any advanced system, then it is automatically by default, it's a part of the software, and software calculates this Nyquist distance for you, and it directly shows you that you should go with this step size. Okay. So once you capture this kind of a Z-step image, see the Z-step image looks like this. Let's say here this is a Z-series image of pollen. Okay, so from top to bottom the pollen looks like this, and these are the different sections of the pollen from top to bottom. And after imaging, you have to do the 3D reconstruction, so you can see the entire 3D image of the pollen. Okay, so here you can see how the pollen looks like in the 3D image. Okay, so this is known as 3D reconstruction. So first you take the Z section images, okay, and after that you have to merge the Z sections to get the 3D reconstruction image like this. So coming to the 4D imaging, or the fourth dimensional imaging. So what is the fourth dimension? So generally the time is the fourth dimension. Okay, so here we will see about time-lapse imaging and live cell imaging. So live cell imaging is the imaging of live cells under controlled environmental conditions such as temperature, CO2, oxygen, humidity. Okay, so let's say if you are imaging cells in the live condition, then you should maintain this kind of environmental parameters such as temperature, generally 37 degrees maintained while working with the cell lines. Then CO2, oxygen and humidity. And if you see for live cell imaging, the microscope has this kind of a environmental chamber. You can see a kind of a box on top of this microscope, which is made up of, which is a transparent box made up of acrylic or the glass. So it is transparent. But inside this box, there are some assemblies to maintain the specific oxygen level, carbon dioxide level, temperature, and humidity. So here you can see the temperature module, carbon dioxide module, oxygen module. So this hardware actually maintain the specific temperature or the humidity or the oxygen and carbon dioxide levels inside this chamber, and you will put your cells or the samples on the stage. So the live cell imaging microscope looks like this, which has a closed chamber to maintain the environmental parameters. Many times, live cell imaging is combined with time-lapse imaging to study the dynamics of cellular processes. Okay, so most of the time, the live cell imaging is combined with the time-lapse imaging. Okay, now there are two types of time-lapse imaging. One is the slow time-lapse imaging, or the other one is the fast time-lapse imaging. So here you can see, let's say you have to determine the time interval for time-lapse imaging. Okay, how you can determine the time interval? So let's say if your objective is to study the process of cell division, then first you should figure out that how long the cell division takes. So generally, let's say in human cells, the cell division process takes one hour. Okay, to segregate the chromosomes and for the cell division. So if you want to image such kind of a slow process which takes 1 hour to complete, you may image at every 5 minutes time interval. Okay, you do not need to image very fast, like at least every 200 millisecond you need to image. Okay, if you go for 200 millisecond imaging, then you will unnecessarily bleach your samples. Okay, so you have to decide the time interval that for such kind of a slow process you can image at every 5 minutes or every 10 minutes. Okay, in 3D. So every 5 minutes or every 10 minutes you will take the three-dimensional image of the cell. Okay, and you can study the process of cell division. But let's say your objective is to capture some fast dynamic processes, such as how long the transcription factor binds to DNA. Okay, so that is kind of a very transcription factor binding to DNA is a very fast process, and it happens in the time scales of seconds. So for that, you need to image at every 200 milliseconds. So here you can see how fast these single molecules of transcription factors they bind to chromatin. Okay, they are very fast, and that's why you have to image at every 200 milliseconds. If you image this kind of a sample with 5 minute time interval, then I think the entire dynamic information is lost. You cannot get any dynamic information because it's so fast. You have to image it in a fast way, like at every 200 milliseconds. But for the process like cell division and all, which takes hours, you can image at every 5 or 10 minute time interval. So this is known as time-lapse imaging and live cell imaging. And this is the fourth dimension. Now, there are several practical considerations for live cell imaging or time-lapse imaging. So for long-term time-lapse imaging, for minutes to hours to days, Z-drift correction, autofocus or perfect focus modules are required to correct for the Z-drift. So what happens? So let's say, suppose if you want to observe the entire embryonic development. Okay, so for observing the entire embryonic development, it takes several hours to days. Okay, so for such kind of long-term time-lapse imaging, let's say what will happen if you have focused your cells here and you have set up entire microscope to capture the time-lapse images at every, let's say, 30 minutes. Okay, for 2 days. Now what will happen? There are always some vibrations from the building or vibrations from the surroundings. Okay, so because of that, the focus may be lost. So maybe after 30 minutes, after 1 hour, when you will see your sample, you will see that your cells or the samples are not in focus. Okay, so this is known as the focus drift or the Z drift because of the building vibrations or some kind of vibrations from the surroundings. Okay, the distance between the objective and the sample may change, and that leads to this kind of a Z drift. So for that, several microscope manufacturers offers Z drift correction modules, or they are known as autofocus modules or the perfect focus module. So what this model does, before imaging at every, let's say 30 minutes or 1 hour, first the microscope will automatically check the focus of the sample. If the sample is slightly defocused, it will automatically focus the sample again before taking the Z-stack image of the sample. So those kind of focus correction modules are generally commercially available, but your microscope should have that module before you plan this kind of a long-term time-lapse imaging experiments. The Z drift occurs due to the smallest vibration around the microscope or the building. And that's why the preferred location for installing a microscope for time-lapse imaging is the ground floor of the building to reduce the vibration. Because if you put your microscope on the 10th floor of the building vibrations will be more. It will lead to frequent defocusing of your sample. But if the microscope is installed on the ground floor or maybe in the basement, then the building vibrations will be less. So the Z drift correction problems will be reduced. So coming to the fifth dimension. So what is the fifth dimension? So fifth dimension is the multicolor. Okay. So we learned about three-dimensional imaging. So now the three-dimensional imaging, you took the entire image of the cell in 3 dimension in one color. But it is possible that you also need that information in multicolor. Like one protein is labeled with magenta color, other protein is labeled with green color, and let the nucleus is labeled with the DAPI in yellow color. Okay, so you need to do multicolor imaging in 3 dimension. Okay, so that will here you can see I have shown the montage of the Z-stack. So see, initially in this channel, yellow channel, the cells were defocused. Then these are all Z-stack images. The cells are perfectly focused somewhere in this area in the frame number 17 and 19. And again, they will defocus. So I have collected the entire Z volume of the cell in 3 different colors, and then here I have shown the merged images of these three. So this is the maximum intensity projection of all these images. This is the maximum intensity projection image of all these images. This is the maximum intensity projection of all these images. So see, in this experiment my aim was to count the number of mRNA per cell. So these individual dots that you are seeing, those are nothing but the individual mRNA within the cell, and I have to count this number of mRNA per cell. Now in this case, let's say if I do undersampling, then what will happen? Let's say this cell has, let's say 10 mRNAs or the 10 spots, but if I haven't taken the proper Z steps and I have done the undersampling, then this cell will show me only 7 dots instead of 10. And if I do the oversampling, it will not give me 12 dots because it has only 10 dots. So it's not possible that the cell will show me 12 dots with oversampling. But only thing is I will unnecessarily bleach my samples more by doing oversampling, and I will also increase the file size. So that's why for extracting such kind of a quantitative information from biological images, you have to be very precise about determining the Z steps and the size of the Z steps. Okay, so the fifth dimension is the multiple colors. Now what is the sixth dimension? So six dimensions is multi-positions. So why we call it six-dimensional imaging? So six-dimensional imaging means you will image the cell in three-dimensional XYZ plus time series plus multicolor plus multi-position. Okay, so let's say you want to image this entire tissue section. Okay, now under the high magnification lens, you cannot see the entire tissue altogether. Okay, so what you will, under the high magnification, you can see only the small part of the tissue. Okay, so what you will need, you will ask the microscope to go to multiple positions and take the image at different positions. Okay, and eventually the microscope software will combine those individual images in this kind of a tile format to show you the entire morphology of the tissue. This is same like you must have played with jigsaw puzzles, right? So to see the entire image, you have to arrange them in proper way so that you can see the entire picture. So same thing, let's say this is the entire field you wanted to image, but under the high magnification you cannot see the entire field altogether. So first you will image this small square like this, and then you will take this second image, and this each image is a Z stacking image, the three-dimensional image and multicolor image. So each of this square is a 4D image, and then uh, it's a 4D image, and then you combine this kind of a all the image in one picture to make the one larger image. Okay, so this is known as 6D imaging. The sixth dimension is the position. However, here the time series is not included. So this is actually the 5-dimensional image, because the time series, this is not a time-lapse movie you are seeing here. It's a static image. So that's why it's a 5D image. But let's say you take this image at every half an hour and make a time series, then it is known as the 6-dimensional image. Okay. So with this, let us end today's class. Our next class, we will learn about how to study protein dynamics using FRAP, FLIP, and FRACT. Thank you.